the enteric nervous system does not have a unique connection to the, directly it is not connected to the central nervous system, while under the influence of parasympathetic preganglionic nerves, release of transmitters usually is dominated by local control. So even though it is not what you call as the independent out of the CNS, through the peripheral nervous systems, they are controlled by the central nervous system. That's why the coordination of contraction and relaxation at local level would be expected for regulation of the peristaltic waves in the intestine. So here the peristaltic waves of the intestine are both controlled by the what you call as the inhibitory and excitatory, or contraction and relaxation, both type of the actions of the intestine. And here you can see that the nervous system, especially the enteric nervous system, is little bit what you call as complicated, and varieties of the receptors are going to act on the varieties of the cells, especially on the enteric nervous system. The PAN means premier calcitonin gene related peptide, CGRP. Here the action of different drugs is mentioned, cisapride, metoclopramide, etc., which are going to inhibit the 5-HT3 type of the receptor. So what happens ultimately, it is going to cause the inhibitory enteric nervous system interneurons, whereas the nitric oxide is a receptor and it's going to cause the inhibitory in nature. So ultimately the muscles are relaxed due to the action of muscarinic 3 type of the receptors, and also the 5-HT3 receptors are being acted with ondansetron, a 5-HT3, and bring the antiemetic type of the action. And here also the 5-HT3 type of the receptors are present in the central nervous system. So ultimately the action is elicited by the different type of the receptors. Here you can see M3 type of the cholinergic receptors, and apart from this one, 5-HT3 and 5-HT4 type of the receptors are also present. And the next important thing is the neurohumoral transmission. So it's one of the important terminology, especially in the nervous, this autonomic nervous system. What's the meaning of the neurohumoral transmission is, humor means it's a chemical, means chemical mediated nerve transmission is the neurohumoral transmission. So when this particular neurohumoral transmission gained importance is in the 20th century, when Lewandowski and Langley, these are the four important scientists in the in the end of 18th century and beginning of the 19th century, this started developing to the greater extent. And they noted the independently and similarity between the effects of injection of extracts of adrenal gland and stimulation of the sympathetic nerves. In case of the external stimuli as well as the internal stimuli to the experiment animals, especially the Lewandowski and Langley. These are the two important scientists. They found the evidence for the neurohumoral transmission, chemical mediated neurotransmission. They know. Then there are varieties of the steps. Please remember all these steps, especially in case of the neurotransmission. The sequence of events involved in neurotransmission is of particular importance pharmacologically, because the actions of large number of drugs are related directly to the individual steps. Why we have to remember the individual steps in case of neurotransmission or neurohumoral transmission is to meddle with them. So suppose we can stop the transmission, we can increase the transmission rate, or we can also decrease the transmission rate. So these are all mediated by the neurohumoral transmission, and if we know very well about the neurohumoral transmission, then we can find many drugs for the further pharmacological action. So there are two terminologies here. Please remember this thing. Axonal conduction is reserved for the passage of an impulse along an axon of the muscle fiber or muscle fiber. Axonal conduction is reserved for the passage of impulse along with the axon. You know what is an axon? It's an elongated nerve body. Then junctional transmission refers to the passage of an impulse across a synaptic or neuroeffector junction. In case of the synaptic junction, in case of the sympathetic nervous system, or neuroeffector junction, especially in case of the cholinergic nervous system. So please remember, the axonal conduction and junctional transmission are the two important processes that is involved in case of the neurohumoral transmission. The first one, that's the axonal conduction. Hodgkin and Huxley in 1952, they found the evidence for axonal conduction. At rest, the interior of typical mammalian axon is approximately minus 70 millivolts, or else it is also known as resting membrane potential. Resting membrane potential. During the rest, the membrane potential will be minus 70 millivolt when compared to the exterior. The resting potential is essentially a diffusion potential based chiefly on 40-fold higher concentration of the potassium in the exoplasm. So in case of the exoplasm, there is higher concentration of the potassium. Then when compared to the exterior, more concentration of potassium are present in the exoplasm, and hence there is negativity inside the exoplasm. And sodium and chloride are present higher concentration in the extracellular fluid than the exoplasm. The external membranes at rest is considerably less permeable to the ions, hence their contribution to the resting membrane potential is small. So the ultimate aim of telling all these things is the resting membrane potential is minus 70 millivolts, especially this is maintained by the potassium channels and also the chloride channels, whereas the sodium and chloride play very less role in case of maintaining the resting membrane potential. What is the resting membrane potential means? Whenever there are no impulses or stimulations, the membrane is electrically depolarized so that the resting membrane, if you measure the electrical activity, then it will be minus 70 millivolt. These ionic gradients, especially the potassium is higher inside during the rest phase, potassium is higher inside the exoplasm and the sodium is higher outside the exoplasm, is mediated by energy dependent transport or pump mechanism, which involves the ATP, adenosine triphosphate, activated by sodium at inner and potassium at the outer surface of the membrane. Here, the resting membrane potential is maintained by sodium and potassium pumps. The resting membrane potential is maintained by the sodium and potassium pumps, whereas the sodium is present outside the exoplasm, whereas the sodium is at the inner and the potassium at the outer side of the membrane, continuously pumping the sodium and potassium respectively. In response to depolarization to a threshold level, action potential or nerve impulse is initiated at a local region of the membrane. So whenever there is action potential is initiated, then depolarization occurs, then the neurotransmission occurs. The action potential consists of two phases. So what's an action potential? It is the electrical disturbance which is maybe originated anywhere in the nerve terminal. So the resting membrane potential is disturbed and it is going to overshoot. Usually it turns towards the positive side. The action potential consists of two phases. The initial phase 1 is caused by rapid increase in the permeability of the sodium through voltage sensitive sodium channels. So, whenever what is an action potential is disturbance of a nerve or in the form of the stimuli. So, what happens? This is going to stimulate the sodium channels. So, in the initial phase, the sodium will be coming inside the exoplasm. Then the result is inward movement of the sodium and a rapid depolarization from the resting membrane potential, which continues as a positive overshoot. Means the resting membrane potential is minus 70 millivolt. Sodium is having more and more positive charges, so the threshold is going to decrease. So the minus 70 millivolts becomes minus 30, then minus 10, minus 1 and 0, then it's overshoots. So that's about the two phases. The phase 1 is consisting of inward movement of the sodium channel, whereas the second phase results from a rapid inactivation of the sodium channel and delayed opening up of the potassium channel, which permits outward movement of the potassium to terminate the depolarization. So here, whenever the action potential arises at any nerve cells or stimuli, then there is inward movement of the sodium channel because of the sodium channel opening, and further the sodium channel is slowly closed because of the opening up of the potassium channel, whereas the potassium channel opens the gate of the potassium channel and there will be inward movement of the potassium to terminate the depolarization. Inactivation appears to involve a voltage-sensitive conformational change, which a hydrophobic peptide loop physically occludes the open the channel in the cytoplasmic side. So the voltage sensitive, all these channels, especially sodium and potassium channels, are voltage sensitive. If the voltage disturbance is there, the channels are going to open or close. And although not important in axonal conduction, calcium channels are also there in other tissues, especially heart, to contribute to the action potential by prolonging the depolarization by an inward movement of the calcium, especially in case of the heart. Apart from this sodium and potassium channels, it is also containing apart from sodium and potassium channel, it's also containing the calcium channels, and these play a very important role in the propagation of the action potential. This influx of calcium also serves as a stimulus to initiate the intracellular events. The transmembrane ionic currents around the axon will produce local circuit disturbance. So, as a result of which localized changes, membrane potential, adjacent resting channels in the axons are activated and the excitation of adjacent portions of the axonal membrane occurs. Usually excitation because of the inward movement of the sodium, the excitation occurs, and this brings about the propagation of action potential without decrement along the axon. So, because this is a saltatory or jumping type of the response, so the propagation of action potential, suppose action potential means any nerve stimuli, it is propagated throughout the length of the nerve due to without any decrement in nature due to the depolarization and repolarization. The pufferfish poison. The pufferfish is one of the most poisonous fish, especially in the water. The tetrodotoxin is secreted by the pufferfish. It's a very potent poison and it's a close congener found in some shellfish. Saxitoxin respectively block axonal conduction. So, it's very interesting that some fish varieties, especially the puffer fish, it secretes the tetrodotoxin and saxitoxins and block axonal conduction. How it is going to block? Especially the tetrodotoxin is going to block the voltage sensitive sodium channel and prevent the increase in permeability to sodium associated with rising pace of the action potential. So, whenever the tetrodotoxin is consumed by the human being or any other animal, like the carnivore animals, then the action potential generation itself is stopped and they prevent the permeability to sodium channel is totally it is decreased and there is no propagation of the action potential. And here you can see this is a nerve fiber. So this is the cell body of the soma what we can already see, and this is the nucleus of that particular cell body, and here you can see that the action potential will be propagated which is generated in the nerve cell body. So myelinated axon, it's the myelinated axon usually. Then the thing is, this is the node of Pranaver, the junction in between these two cells, and it is a myelin sheath. Whereas the action potential is generated due to the disturbance of the electrical current, then it's propagated throughout the nerve. So in case of unmyelinated axons, it's also the action potential is also propagated, whereas it is jumping in nature, whereas it is continuous in nature in case of unmyelinated axons. So this is interesting. The resting membrane potential is minus 70 millivolt. It is maintained by two channels. One is sodium channels and the potassium ion channels. Whenever the tissue is under rest, the resting membrane potential will be minus 70 millivolts. So as soon as the action potential arises here, there will be massive inward movement of the sodiums. So this reduces the threshold to minus 55 millivolt. Then the depolarization occurs. There will be continuous movement of the sodium ions inside the neuron cell, and it reaches to peak, and at that time the potassium channel opens. Then the repolarization occurs, and because when it reaches the what you call plateau, then because of the potassium ion pumps, once again lot of potassium ions are pushed inside. It causes the hyperpolarization, and when it comes to the normal, that's known as the resting membrane potential. Once again minus 70 millivolt. It downshoots to 100 millivolts, of course, then it comes to the normal. This is a very regular cycle of an action potential. Action potential. This is containing the stimuli comes to any of the nerve terminals, then there is sudden opening up of the fast opening up of the sodium channels, then it causes the initiation of the generation of action potential. This shoots to the positive side, the depolarization occurs, and immediately after the threshold, the potassium channel is going to be opened, and this causes the repolarization ions, whereas the sodium ions are going out and potassium ions are coming in. Then this, because of this movement, it goes to more and more negativity and hyperpolarization occurs, and once again it's coming to the resting membrane potential due to the potassium and sodium pump action.